Good morning. Today I am going to talk on an article with the title "A Lab on a Chip for the Concurrent Electrochemical Detection of SARS-CoV-2 RNA and Anti-SARS-CoV-2 Antibodies in Saliva and Plasma." This article was published in the journal Nature Biomedical Engineering with an impact factor of 26.6 in 2020. So as I was discussing about the EFIRM detection of salivary biomarkers, the same concept is used here, which is a lab on a chip for electrochemical detection of both the viral RNA as well as the host antibody in the saliva as well as plasma. And this study was conducted in the Weiss Institute of Biologically Inspired Engineering, Harvard University, as well as the Institute of Medical Engineering in Massachusetts, Institute of Technology, Cambridge. So we know molecular diagnostics for detection of pathogen RNA and the serological assays for assessment of host antibody responses are complementary. So that provides critical information to respond to disease outbreaks, assess vaccination status, and manage patient care and risks. So all these things can be managed with a simple diagnostic detection of the pathogen RNA as well as the antibodies from the host. So when we talk about the lab on a chip, it mainly used microfluidics. Microfluidics is the small channels are there in a chip where when we place a sample, it just moves to the different wells where the different detectors are present to find the biomarkers. So it is mainly used for cell-cell interaction analysis, genetic analysis, drug discovery and diagnostics. Here we are going to talk about the diagnostic part where it detects the RNA as well as the antibody of the host. So this is how it is, the chip will be present and where they add the sample through these channels. This is called as the microfluidic technique, where it reaches the particular area where it has to go and cause the interaction with the reporters. So the novelty in this study is, despite the advances to make SARS-CoV-2 tests as widely as possible, a lab on a chip platform for point of care use that can detect both SARS-CoV-2 and RNA and the serological markers was not available. So this study aimed to find a rapid, accurate and frequent detection of RNA of SARS-CoV-2 and serological host antibodies to the virus. So this aim of the study was to design a lab on a chip. So how did they do? They did a chip preparation where the gold chips were custom manufactured by a tele company using a standard photolithography process with the deposition of 500 nanometer chromium and 100 nanometer gold on a glass wafer. Next, they did a nano composite preparation and activation using amine functional residue, graphene oxide, and the clinical samples were collected with an ethics statement. Like they de-identified clinical saliva samples from and these plasma samples were collected from different areas, and CRISPR-based RNA assay with fluorescent reporter was used. So CRISPR-based RNA assay requires the selection of both lamp-less loop-mediated isothermal amplification. So isothermal here means of the same temperature. Usually we increase the temperature, decrease the temperature for amplification. Here they use the isothermal amplification method, and for that they have the primers and the guiding RNAs to detect the lab antigens. And chip functionalization for the CRISPR-based RNA assay with electrochemical reporter. So as I said, there should be a reporter primer also. For CRISPR sensors, custom synthesized amine-terminated peptide nucleic acid, we call it as PNA, was used. The PNA is a synthetic analog of a DNA with a backbone utilizing repeating units of N-glycine linked through amide bonds. So the PNA contains the same 4 nucleotides as a DNA, but connected through methylene bridge and the carbonyl group to the central amine of a peptide backbone. So this is about the reporter PNA. And the CRISPR-based RNA assay with electrochemical reporter. The reporter peptide sequence for CRISPR was base BCSAs was ssDNA, which is conjugated to a biotin linked to polystreptavid HRP. As I said, it has to have a sensor to detect whether that is present or not. And the serology ELISA assay was also done. So they did a serology ELISA assay. This is a lab procedure, and this was a lab on a chip. So ELISA assays were optimized in 96 well plate, and they did all these detection through the ELISA test. And electrochemical serology assay was also conducted to translate the ELISA assays to EC readouts for the S1N and S1 RBD. I will just talk about it in a few minutes. which were diluted to 1 milligram per ml in the PBS buffer and spotted in 3 electrodes of ECG. And the multiplex electrochemical serology and CRISPR-based RNA detection was done. And microfluidic chip for multiplex electrochemical serology and CRISPR-based RNA detection, where a microfluidic chip for a point-of-care diagnostic that integrated multiplex chip was designed using AutoCAD software and printed on a 3D printer in grey. So the results are, how did they design a microfluidic electrochemical lab on a chip platform? So this was the parts of the microfluidic chip, where it has a resistor, the resistor holder, and this is the microfluidic chip, which has a PES membrane here, and there are wells for the samples, and there are places where one is a sample preparation reservoir for saliva, and the next one is a reaction chamber with a PES membrane. Third is the lamp reservoir, which is the loop amplification for isothermal amplification. This is a CRISPR and a reservoir and the integrated multiplex DC sensor. This is called as a multiplex, where we can